Guys, there was some concern around the fact that coronavirus is being mutated and vaccine not working. So just came across this article. It actually is this product is going through preclinical trials. It's a protein nanoparticle vaccine. It's very interesting, and I'm gonna try to make this video as interesting as possible. So, so the way they designed this nanoparticle vaccine is this vaccine display 60 copies of SARS-CoV-2 spike glycoprotein receptor binding domain and a highly immunogenic array and induce neutralizing so that induces neutralizing antibody titer roughly tenfold higher than the prefusion stabilized S ectodomain trimer despite a more than fivefold lower dose. So I guess pre-fusion stabilized as ectodomain trimer. I did some research and I found out that um, so here it is. Coronavirus are decorated with a spike glycoproteins that binds to host cell receptor and mediates cell entry via fusion of the host and viral membranes. So, as proteins are trimeric class 1 fusion proteins that are expressed as a single polypeptide that is subsequently cleaved into S1 and S2 subunit by cellular proteases, a stabilized perfusion conformation of class 1 fusion proteins is desirable for vaccine development because this conformation is found on infectious virons and displays most or all of the neutralizing epitopes that can be targeted by antibodies to prevent the entry process. Um, we and others have observed that pre-fusion stabilization tend to increase the recombinant uh, expression of viral glycoprotein, possibly by preventing triggering or misfolding that results from a tendency to adopt the more stable post-fusion structure. For example, structure-based design of pre-fusion stabilized MERS-CoV and SARS-CoV spike ectodomain resulted in um, homogeneous preparation of pre-fusion spikes and greatly increased yield. These variants contain two consecutive proline substitution in the S2 subunit in a turn between the central helix and heptad repeat one that must transition to a single elongated alpha helix in the post-fusion conformation. Pre-fusion stabilized spike variants are also superior immunogens to wild-type spike ectodomains and have been used to determine high-resolution spike structure by cryo-EM. Importantly, the successful transplantation of this double proline substitution into the SARS-CoV-2 allowed for the rapid determination of high resolution chiral EM structure and accelerated development of vaccine candidates. Anyway, so the long story short, they have developed this pre-fusion conformation of class 1 fusion protein, but this right here, I'm not gonna go into much detail. It's a waste of time at this point, but I guess it's all about stabilization of uh, a structure-based design to generate a pre-fusion spike protein that expresses at higher level and is more stable than our original S2P construct. We analyze the SARS structure and design substitution based upon knowledge of class 1 fusion protein function and general protein stability principles. These strategies include the introduction of disulfide bond to prevent conformational changes during the pre to post-fusion transition, salt bridges to neutralize charge imbalances, hydrophobic residues to fill internal cavities, and prolines to cap helices or stabilize loops in the pre-fusion state. We clone 100 single S2P variants and characterize their relative expression level, and for those that express well, we characterize their monodispersity, thermostability, and quaternary structure. Given that the S2 subunit undergoes large-scale refolding during the pre to post fusion transition, we exclusively focus our effort on stabilizing S2. Substitution of each category were identified that increase expression while maintaining the prefusion conformation. Overall, 26 out of 100 single substitution variants had higher expression than S2P. One common strategy to stabilize class 1 fusion proteins such as the spike is to covalently link a region that undergoes a conformational change to a region that doesn't via disulfide bond. For instance, attachment and entry are essential for the viral life cycle, making the S protein a primary target uh, of neutralizing antibodies and a critical vaccine antigen. Binding of the RBD to ACE2 triggers S1 dissociation, allowing for a large rearrangement of S2 as it transitions from a meta-stable pre-fusion conformation to a highly stable post-fusion. Got it. Okay, got it. In a highly immunogenic array, and induced neutralizing antibodies titer roughly tenfold than prefusion stabilized as ectodermal despite a more than fivefold lower dose. These results highlight the utility of robust antigen display platforms for inducing potent neutralizing antibody responses and have launched